Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjon classes. Dear student, we are in a continuation of the chapter that is respiration in plant. In previous class, we have already talked about uh, introductory part of this chapter as well as the one important method in the type of respiration that is a glycolysis. Now, as we have discussed in the glycolysis, the glucose get converted into the two molecules of pyruvic acid, and this pyruvic acid is of three carbon compound. Now, what will happen to that pyruvic acid that we have to discuss here? And in that, as we know, and if you talk about a fate of pyruvic acid, so fate of pyruvic acid help us to understand that how this pyruvic acid is going to break broken up. So it is known that the fate of pyruvic acid, it is decided based on presence or absence of O2. And if we talk about a presence of absence of O2, it is known that if the presence of O2 is there, then in the presence of O2, pyruvic acid get converted into acetyl coenzyme A, and in absence of O2, it is carried out a fermentation. And we will discuss this fermentation in detail in later part. But now we are going to talk about this thing in the presence of O2, and that is known as link reaction. So first we will discuss a link reaction, and from acetyl coenzyme A, Krebs cycle will work. So we will discuss about Krebs cycle also. So this is the thing. So first we are going to talk about aerobic respiration, that is in the presence of O2. And in glycolysis, as we have discussed, that is also a part of this aerobic respiration. So if we go on discussing this further, first thing in aerobic respiration is that it takes place within the mitochondria. Means whatever the breakdown in the presence of O2 is going to take place for the pyruvic acid, that is going to happen in the mitochondria. And first, the pyruvic acid, which is synthesized in the cytoplasm, that is transported from the cytoplasm to the mitochondria. And in mitochondria, you will see that the two important events in aerobic respiration take place through which the breakdown of pyruvic acid occur. Now, in this, the first thing is that we will talk about the complete oxidation of pyruvic acid by the stepwise removal of all the hydrogen atoms and ultimately leaving or removing the three molecules of CO2. Means whatever we are going to study here, that we are going to talk about the link reaction and Krebs cycle. Link reaction and Krebs cycle will help us to understand how the pyruvic acid undergo oxidation and the stepwise removal of hydrogen atom occur from them, and how the three molecules of CO2 are released. Same way, after the Krebs cycle, we will see that the passing of electron removed as a part of hydrogen atom to a molecular oxygen through the simultaneous synthesis of ATP. We will understand that in a link reaction and Krebs cycle, NADH2 and FADH2 are going to be synthesized. Now, NADH2 and FADH2 are one type of energy carriers. They are reduced coenzymes. It is known that NADH2 has 3 ATP and FADH2 has a 2 ATP. Means NADH2 is equal to 3 ATP and FADH2 is equal to 2 ATP. But this NADH2 and FADH2 gives this much ATP when ETS occur, and oxidative phosphorylation occur. So this passing of electron and removal of hydrogen atom from the NADH2 and FADH2, and ultimately passing of electron to the molecular oxygen, and then the synthesis of ATP, that all occur through the ETS and oxidative phosphorylation. That also we will discuss here in the later part. Okay, so So now we have to discuss this in much detail. Okay, so this is the what we are going to understand. So the first event, that is a Krebs cycle and the link reaction that takes place in the matrix of mitochondria, and the second event, that is the ETS, that takes place on the inner membrane of the mitochondria, as we have discussed, and the oxidative phosphorylation also takes place on the crystal. So this is the thing, you can see this is the yellow colored part is the mitochondrial matrix, where this Krebs cycle and link reaction will take place. Here link reaction and Krebs cycle will take place. Whereas on the inner mitochondrial membrane, here you will see the presence of what ETS, and here F0 F1 complex are located like this on the crystal, that we will carry out a oxidative phosphorylation. Okay, now first we are going to talk about the link reaction or oxidative decarboxylation of pyruvic acid. So if we talk about the first link reaction, this link reaction is also called as gateway reaction, or it is also known as link reaction or gateway reaction or transition reaction. So link reaction, gateway reaction or transition reaction are there. Now in this what happens, the pyruvic acid further break up in the presence of oxygen. Actually directly oxygen is not involved, but indirectly the involvement is there, and ultimately this pyruvic acid get converted into acetyl coenzyme A. So this link reaction, gateway reaction, transition reaction, or it is also called as pyruvate dehydrogenase complex. Now what happens in this, let's understand. First we have a pyruvic acid. Now this pyruvic acid is of 3 carbon compound. Now what happens, first the decarboxylation takes place and CO2 get released. Now CO2 is of 1 carbon. So from pyruvic acid, if 1 carbon get removed, it is going to form a acetate moiety or acetate molecule. In this acetate molecule, coenzyme A get added along with the release of hydrogen. That hydrogen are accepted by NAD plus, and this NAD get converted into NADH2. Okay, and ultimately by conversion of this and addition of coenzyme A, acetate get converted into acetyl coenzyme A. This acetyl coenzyme A is of 2 carbon compound. This acetate is also of 2 carbon compound. So, but when we have a one pyruvic acid, one CO2 will be released, one NADH2 will be released, one coenzyme A get added, and one acetyl coenzyme A get formed. But as we know, glycolysis is going to produce a 2 pyruvic acid. So, from 2 pyruvic acid, 2 link reaction will take place. So, if 2 link reaction will take place, 2 CO2 get released, 2 acetate molecule will be formed, 2 NADH2 will be formed, and 2 coenzyme A will be added. So, ultimately, 2 molecule of acetyl coenzyme A are formed. And if we have 2 acetyl coenzyme, 2 Krebs cycle is going to happen further. Okay, so this is the what a 2 link reaction will take place when there is a 2 pyruvic acid from the 1 glucose molecule. So in 2 link reaction, you will see that there is a formation of 2 NADH2 molecule and release of 2 CO2. But if there is a 1 link reaction, 1 CO2 and 1 NADH2 get released or synthesized. Now to carry out this whole reaction, pyruvate dehydrogenase complex work, and pyruvate dehydrogenase complex consists of enzyme plus coenzymes. They have PDH enzyme plus. So here pyruvate dehydrogenase complex is a PDH enzyme, that is pyruvate dehydrogenase enzyme, plus several coenzymes are involved here, and the coenzymes like lipoic acid, thymine pyrophosphate, then lipoic acid, thymine pyrophosphate, NAD plus, coenzyme A. Okay, these all are the particular type of coenzymes. Along with that, some cofactors are also involved to carry out this reaction, and cofactors like magnesium is involved here. Okay, so this is the what pyruvate dehydrogenase complex, जिसमें ये PDH एंजाइम कोएंजाइम एंड कोफैक्टर यहाँ पे इन्वॉल्व होते हैं और वो पायरुविक एसिड को असिटाइल कोएंजाइम में कन्वर्ट कर पाते हैं. Okay, so that is the what a thing we can say, and ultimately two molecule of acetyl coenzyme will be formed in this pyruvate dehydrogenase complex or link reaction, and this acetyl coenzyme will be further used in the Krebs cycle. So we have to discuss how this happens, where it happens. So this is what a link reaction. So as we know, this link reaction occurs in mitochondrial matrix. As we have discussed in the link reaction, pyruvate, which is found by the glycolytic catabolism of a carbohydrate in the cytosol, after entering the mitochondrial matrix, undergo oxidative decarboxylation by a complex of set of reaction that is catalyzed by a pyruvate dehydrogenase enzyme complex, as we have discussed just now. So the reaction catalyzed by pyruvate dehydrogenase require the participation of several coenzyme, as we have discussed, the NAD plus, coenzyme A, TPP, thymine pyrophosphate, all coenzymes, several coenzymes are involved and cofactors are required. So ultimate if we reaction, we summarize it, pyruvic acid react with the coenzyme A and along with the NAD plus, it will give rise to acetyl coenzyme A plus CO2. NADH2 will be obtained and it take place in the presence of pyruvate dehydrogenase enzyme and which require also magnesium. So ultimately acetyl coenzyme we are going to along with the decarboxylation. Because of this we are calling it a oxidative decarboxylation. So during this process, two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid produced from one glucose molecule during the glycolysis. Okay, so two link reactions are required to produce two NADH2 and two pyruvic acid breakdown. The acetyl coenzyme, which is obtained at last, that enters a cyclic pathway, which is called a TCA cycle or Krebs cycle, and in that this acetyl coenzyme is going to be used. How it is used, how Krebs cycle takes place, we will discuss in a later part in the continuation of this lecture. Before that, first we'll discuss some of the question. The final product of glycolysis, as we have discussed in the previous glycolysis, that a pyruvic acid or pyruvate is the final product of glycolysis. Pyruvate undergo oxidative decarboxylation is obviously it is talking about the pyruvate dehydrogenase complex or a link reaction or a gateway reaction, and that takes place obviously in the mitochondrial matrix. Then the enzyme involved in oxidative decarboxylation of pyruvate is obviously it is a pyruvate dehydrogenase enzyme, that is option 3. Coenzymes involved in oxidative decarboxylation of pyruvic acid is obviously NAD, coenzyme A, FMN. FMN is not involved, so both 1 and 2 become the correct answer. Connecting link between the glycolysis and Krebs cycle. Yes, this is the one of the important question that you should remember, that glycolysis and Krebs cycle are linked by this link reaction. So because of this connecting link between the Krebs cycle and glycolysis is the acetyl coenzyme A, because acetyl coenzyme will be obtained from the pyruvic acid and that undergo a Krebs cycle. So now we are going to discuss here mainly a Krebs cycle or tricarboxylic acid cycle in the continuation of this aerobic respiration. This tricarboxylic acid cycle is also known as citric acid cycle or a Krebs cycle. Why it is known as Krebs cycle? Because it is discovered by Hans Krebs. Citric acid cycle because first stable product is a citric acid. And okay, so this is the thing. So citric acid cycle or tricarboxylic acid cycle and Krebs cycle. So in 1937, English biochemist Sir Hans Krebs postulated the cycle of reactions which describe the oxidation of pyruvate to CO2 and came to known as a citric acid cycle. In 1953, Krebs was awarded a Nobel Prize and the cycle is frequently referred as a Krebs cycle in his honor. So we are going to discuss how this Krebs cycle takes place. So to understand a Krebs cycle, As we know, the after a link reaction, this Krebs cycle is going to take place, and a product of link reaction is a acetyl coenzyme A, which is of two carbon. I am taking a one molecule, and we are going to discuss one Krebs cycle only. No doubt, from one glucose two Krebs cycle will take place, but here I am making you to understand one Krebs cycle, how take place, that will help you to understand detail. Now, in this Krebs cycle, in this Krebs cycle, as we know, the acetyl coenzyme A react with oxaloacetic acid, and oxaloacetic acid is a four carbon compound, whereas acetyl coenzyme A is a two carbon compound. Acetyl coenzyme A react with a oxaloacetic acid, a two carbon compound. Okay, and in this acetyl coenzyme A, that is a two carbon, and oxaloacetic acid, which is a four carbon, that react with each other by the enzyme that is citrate synthase. And citrate synthase is a condensing enzyme that make a condensation or a reaction of the acetyl coenzyme with the oxaloacetic acid and form citric acid. This citric acid is a six carbon stable compound of this one. And to carry out this reaction here, addition of one water molecule take place, and along with that, a coenzyme A from acetyl coenzyme A is going to release. Now this citric acid get converted into cis-aconitic acid, and this cis-aconic acid, when from the citric acid, the water is going to be released, and when water get released here, the enzyme aconitase get used, and aconitase need Fe2 plus for this activity as a cofactor. Further, this cis-aconic acid, by the addition of water, again get converted into the isomer of citric acid, that is a isocitric acid, which is of six carbon, and it is isomer of citric acid. So here also aconitase enzyme get used, and this aconitase also need Fe plus 2. So here this as well as this are the reversible enzymes, are the reversible enzymes. Okay, and this citric acid get converted into isomer isocitric acid by the formation of an intermediate, that is a cis-aconic acid. Now this isocitric acid, which is a six carbon, get converted into oxalosuccinic acid, and this oxalosuccinic acid is also of six carbon in the presence of enzyme isocitrate dehydrogenase. And when isocitrate dehydrogenase will work, H plus get removed, that H plus are accepted by NAD plus and get converted into NADH2. Okay, so this way the isocitric acid get converted into oxalosuccinic acid, and this oxalosuccinic acid further converted into alpha ketoglutaric acid by the enzyme oxalosuccinate decarboxylase. So here oxalosuccinate decarboxylase will enzyme will work, and this oxalosuccinate decarboxylase enzyme will work, then CO2 get released, And this oxalosuccinate decarboxylase required Mn2 plus, and in decarboxylation CO2 get released and is converted into alpha ketoglutarate. Alpha ketoglutarate. So this is the oxalosuccinic acid by the release of CO2 get converted into alpha ketoglutarate, and this alpha ketoglutarate is a 5 carbon compound due to the decarboxylation. Now this alpha ketoglutarate or alpha ketoglutaric acid is the only 5 carbon compound in this citric acid cycle. Here further this alpha ketoglutaric acid get converted into succinyl coenzyme A with the help of alpha ketoglutarate dehydrogenase complex. Now when this dehydrogenase complex will work, here the CO2 get released along with NAD get converted into NADH2 by accepting the H plus, and coenzyme A is also getting added. So ultimately this alpha ketoglutarate dehydrogenase gets converted into succinyl coenzyme A. Succinyl coenzyme A is a 4 carbon compound and alpha ketoglutaric acid is a 5 carbon compound. Due to the CO2 release, the 4 carbon compound is formed. This is the alpha ketoglutarate dehydrogenase complex. So it also needs lipoic acid, thymine pyrophosphate, then coenzyme A and NAD and magnesium to carry out its reaction. And here decarboxylation and dehydrogenation both are taking place. Further, succinyl coenzyme A in the presence of succinate thiokinase enzyme get converted into succinic acid. So here the enzyme which is going to be used, that is a succinate thiokinase. Now during this the coenzyme may get released from this, and along with this the GDP get converted into GTP. So GDP react with inorganic phosphate and converted into GTP, and this GTP when break it converted into GDP. But when GTP breaks, it releases its phosphate, and this phosphate is accepted by ADP. So this get ADP get converted into ATP. So GTP will form first, and GTP when it will break, it will form a ATP. So either GTP or ATP synthesis occur during this step. Further, this succinyl CoA acid, which is a 4 carbon compound, that converted into fumarate or fumaric acid by the enzyme succinate dehydrogenase. And during this, when succinate dehydrogen enzyme will work, the H plus are getting released, and this H plus are received by FAD plus, so it is converted into FADH2. So here FADH2 synthesis occur due to the dehydrogenation of succinic acid, and fumaric acid will help, and here succinate dehydrogenase enzyme will work. Then this fumaric acid get converted into malic acid. This fumaric acid get converted into malic acid, and this fumaric acid get converted into malic acid by the enzyme fumarase. And when fumarase will work, the H2O get added in this reaction. And this malic acid, further in the presence of malate dehydrogenase, malate dehydrogenase, the H plus get released from the malic acid, and that is accepted by NAD plus, and this NAD plus get converted into NADH2. Okay, NADH2. Okay, so this is the what a ultimate reaction, and then the oxaloacetic acid will be formed again. So this is the whole Krebs cycle how it takes place. And in this Krebs cycle, you will see the dehydrogenation and decarboxylation. Wherever the dehydrogenation will take place, there the NADH2 or FADH2 is going to be synthesized. And the decarboxylation is also going to take place. So here you can see this is a one NADH2, this is another NADH2, this is a third NADH2, and this is the FADH2. This all are synthesized during the dehydrogenation, and the decarboxylation also take place here, as well as we can say here, and one in the leak reaction. So from pyruvic acid, this three times a decarboxylation is going to take place. Okay, so this is the what particularly about the Krebs cycle. So now, by understanding this Krebs cycle, what we understood here. So first, we have to understand the ATP calculation. Before ATP calculation, let's note some of the important points. And an important point it is that Krebs cycle that occur inside mitochondrial matrix occur inside mitochondrial matrix of eukaryotic cell and cytoplasm of prokaryotic cell. So this Krebs cycle takes place in mitochondrial matrix of eukaryotic cell and cytoplasm of the prokaryotic cell. As we have discussed in Krebs cycle, one turn of Krebs cycle involve four dehydrogenation, four dehydrogenation, two decarboxylation, and one substrate level phosphorylation. One substrate level phosphorylation. These all are going to take place in one turn of Krebs cycle. Okay. Then the oxaloacetic acid is considered as first. See, before that, see, one substrate level phosphorylation means it is talking about ATP synthesis. Two decarboxylation means two CO2 release, and four dehydrogenation means NADH2 and FADH2 synthesis. So three NADH2 and one FADH2 are getting synthesized. Apart from this, oxaloacetic acid is considered as the first member of the cycle. Okay. Now, further if we talk, then we remember that all enzymes of Krebs cycle, are located inside mitochondrial matrix. Because of this, the Krebs cycle takes place in mitochondrial matrix. But apart from all enzymes which are located in mitochondrial matrix, there is one exception, and it is known as except succinate dehydrogenase, or SDH enzyme. So this SDH enzyme is a marker enzyme, and this marker enzyme is located in inner mitochondrial membrane. So all enzymes are located in the mitochondrial matrix except this one that is located in the inner mitochondrial membrane, and that is succinate dehydrogenase enzyme, where we have seen the synthesis of FADH2. Okay, so that is the what about the particularly Krebs cycle. Now, if we talk about the ATP calculation, of one Krebs cycle. So in one Krebs cycle, we will see that three NADH2 are synthesized. Now one NADH2 is equal to 3 ATP. So 3 into 3 ATP is equal to 9 ATP. Apart from this, one FADH2 gets synthesized. One FADH2 is equal to 2 ATP, and 2 ATP is equal to, we can say, that is 2 ATP. So total 11 ATP are synthesized. And this 11 ATP are obtained from NADH2 and FADH2. Means NADH2 and FADH2 when break under the LTS, they will give you this 11 ATP. So these are the ATP which are obtained through oxidative phosphorylation. Whereas one ATP is synthesized directly through substrate level phosphorylation. Means the phosphate is released from the substrate and synthesis of ATP takes place. So total 12 ATP are obtained from one Krebs cycle. But if there is a one glucose breakdown is taking place, two pyruvic acid will be formed, two acetyl coenzyme will be formed, and two Krebs cycle will occur. So if two Krebs cycle will occur, this 12 into 2 is going to be 24 ATP. Okay, such a way 24 ATP are going to be produced. Okay, so this is the thing. So all the reactions we have discussed, all the sequences we discussed, just we are revising once again. The sequence of Krebs cycle. First, we have this in the reaction of condensation, where oxaloacetic acid is reacting with the acetyl coenzyme along with the water and forming the citric acid plus coenzyme is released in the presence of citric enzyme. Then we have discussed that the dehydrogenation will occur, and from the citric acid in the presence of aconitase and cis aconitic acid will be formed, and one water is released. Then the hydration takes place again, one water is added and cis aconitic acid get converted into the isocitric acid. So here we should write that addition of one water. Then oxidation first will occur, the isocitric acid in the presence of isocitric dehydrogenation, H plus will remove from isocitric acid that is accepted by NAD and converted into oxalosuccinic acid, and NADH2 is getting synthesized. Then the decarboxylation will occur, oxalosuccinic acid undergo decarboxylation in the presence of oxalosuccinate decarboxylase enzyme, and alpha ketoglutarate acid will be synthesized and CO2 is getting released. Then the alpha ketoglutarate acid further undergo oxidative decarboxylation, that is oxidation, and where alpha ketoglutarate acid react with NAD and coenzyme A in the presence of alpha ketoglutarate dehydrogenase enzyme complex, and succinyl coenzyme will be formed along with the synthesis of NADH2 and CO2 release. Then the succinyl coenzyme A react with the ATP and inorganic phosphate in the presence of succinate thiokinase enzyme, and succinic acid will be released, synthesized along with the ATP synthesis and coenzyme will be released. Then the third oxidation step will take place, and the succinate acid will react with FAD and in the presence of succinate dehydrogenase enzyme, and the fumaric acid will be synthesized, and FADH2 is going to be released. Next, the fumaric acid undergo hydration with the addition of water in the presence of fumarase, form of malic acid. In the last step, the oxidation fourth will take place, and the malic acid will react with NAD in the presence of malic dehydrogenase, and the oxalosuccinic acid synthesized and NADH2 is going to be synthesized. So three NADH2, one FADH2 are synthesized in this whole Krebs cycle. So if we summarize the whole this equation of the oxidation of pyruvic acid in mitochondria, then pyruvic acid reacts with the four NADH2 from the pyruvic acid, means it including both link reaction and the Krebs cycle. Then pyruvic acid reacts with the 4 NADH2 because in a link reaction also one NADH2 is getting synthesized. So 4 NADH2 are used, one FAD is used, two water molecule are used, and ADP and organic phosphate are also involved. So in the mitochondrial matrix, 3 CO2 are released, 4 NADH2 will be synthesized, one FAD has to get synthesized, and one ATP is going to be synthesized. So that is the ultimate summarized reaction. Now we have to understand some of the questions. So isocitrate acid is the substrate for first oxidation of the Krebs cycle, first decarboxylation step in mitochondrial matrix, third oxidation step in aerobic respiration, and second oxidation reaction in mitochondrial matrix. Now we have to select the option among these four. So if we talk isocitrate, isocitrated decarboxylase, isocitric acid is the isomer of citric acid. And further, isocitric acid in the presence of isocitrate dehydrogenase enzyme undergo dehydrogenation and synthesis of NADH to take place. And oxalosuccinic acid is going to be formed. So in this reaction, there is a first oxidation step in Krebs cycle, obviously. Decarboxylation not occurring in that particular step. Third oxidation is not occurring in that step. Second oxidation reaction is also not occurring in this step. So only one is third oxidation reaction. Here they are given the first, third, and fourth. Okay. Third oxidation reaction in my aerobic respiration will be given in third step. Yes, so first NADH in the forming in the glycolysis, second in the link reaction, and third here. So it is the third, obviously. And second oxidation reaction in mitochondrial matrix. Obviously, in mitochondrial matrix is the second oxidation reaction. But in Krebs cycle is the first oxidation. It is a very good question. First, third, and fourth are going to be the correct. Be remember. In the third statement, we are going to look for the NADH to FADH synthesized from the glucose through glycolysis. So in glycolysis, one NADH2, first oxidation, in link reaction, second oxidation, and third oxidation in the which one? The particularly we can say uh, Krebs cycle. And in mitochondrial matrix, the first the Krebs cycle link reaction dehydrogenation, and the second oxidation is this one. But in Krebs cycle, it is the first one. So first, third, and fourth are going to be the correct. Second question, the number of ATPs formed through the SLP and FADH2 oxidation in 2 turns of TC cycle. So if 2 turns of TC cycle, in 1 turn of and in TC cycle, 1 FADH2 and 1 SLP take place. So 1 ATP in the cycle. So number of ATP if want to calculate, then here they are talking about 2 turns of cycle. So in 1 TCA cycle, 1 FADH2 and 1 ATP through substrate level phosphorylation. But if 2 turns are there, then it is going to be 2. So 2 FADH2, and here this is 2 ATP. Now this 2 FADH2, 1 FADH2 gives rise to 2 ATP. So it is going to be 4 ATP plus this 2 ATP. So total is going to give rise to the 6 ATP. Okay. So here 6 is going to be the correct answer. The only 5 carbon intermediate of the TCA cycle is precursor of the following amino acids. So obviously, that particularly it is known that the alpha ketoglutarate acid is a 5 carbon intermediate of TCA cycle and is the precursor of the several amino acids. Identify the correct statement about the succinate dehydrogenation. It involves oxidation of the substrate but not in decarboxylation. FADH2 is getting synthesized when this reaction this enzyme. So obviously the oxidation of the substrate is taking place, but decarboxylation is not taking place. So obviously it is correct. But it is the enzyme of the third oxidation step in TCA cycle. Obviously, in TCA cycle it is the third step. It is the enzyme for the fifth biological oxidation in aerobic respiration. So if we talk about the whole aerobic respiration, first in glycolysis, second in link reaction, third during the isocitrate dehydrogenase enzyme, fourth during the alpha ketoglutarate dehydrogenase enzyme, and fifth during the succinate dehydrogenase enzyme. So it is the correct one. And it is the enzyme of the fourth oxidation step in mitochondrial matrix. Then also it is the correct one. NAD is the coenzyme for this whole enzyme. Here, NAD is not required. FAD is required actually. Okay. So if we talk about ACDE, BCDE, and EBD, the ABCD are the correct one. So the fourth is going to be the correct answer. In Krebs cycle, FAD is involved in the formation of succinic acid, fumaric acid, malic acid, and coenzyme A. So FAD is involved in the formation of their FAD has to use succinate dehydrogenase. So in succinate dehydrogenase work, it converts succinic acid into fumaric acid. So fumaric acid is the product. By the end of the Krebs cycle, most of the energy removed from the glucose molecule has been transferred to NADH2 and FADH2 obviously. Products of cleavage reaction in Krebs cycle. Cleavage reaction is where the succinyl coenzyme get converted into the succinic acid. So here succinyl coenzyme react with ATP. Products, so succinyl coenzyme react with the ADP and coenzyme A is released. Succinic acid is going to be formed. So succinic acid, ATP, and coenzyme are the product. In Krebs cycle, the substrate for final oxidation step in the malic product of following biochemical reaction. So it is the last substrate is the malic acid that undergo dehydration. dehydrogenation, but it is the product of a hydration reaction from the fumaric acid. The number of ATP molecule formed through the substrate level phosphorylation in glycolysis in Krebs cycle during the complete oxidation of one glucose molecule. So in one glucose molecule, if we talk, and so one glucose is that the number of ATP molecules formed through the substrate level phosphorylation. So in glycolysis, we know that 4 ATP are produced, and in Krebs cycle, from one glucose, 2 Krebs cycle will take place. So 2 ATP will be produced. So 4 and 2 is going to be the correct answer. So this is all about what a Krebs cycle, link reaction in Krebs cycle. Okay, now the ETS is remaining how the ATP are 3 ATP are obtained from the NADH2 and 2 ATP obtained from the FADH2. That we are going to understand in the separate lecture in the topic that is the electron transport system. Okay, I hope you understood this Krebs cycle and link reaction very good in a very good manner. Thank you, everyone.